All right. Well, afternoon and evening, everybody. My name is Damon Tai. I am Bio-Rad's curriculum and training specialist for the Western region of the United States. So I'm based out in the San Francisco Bay Area. I'll give you my background just because I think it's always important to know your host's strengths and weaknesses. So I'm a former high school teacher, taught up in Portland, Oregon. And then I moved down to the San Francisco Bay Area to work on the Human Genome Project, i.e. I was really into DNA, not protein stuff. And so a little bit of this isn't quite my wheelhouse, but I'm gonna do my best with it tonight as I can. I moved to Bio-Rad about 10 years ago because when I was at the national labs, I was running into this problem where I would get four-year grads and they'd be really good in interviews. And then I'd put them in basically in the lab space, train them on what I thought they needed to be trained on, and watch them use a pipette and go, oh my God, have you become our dishwasher or something? And so I kind of joined Bio-Rad to help drive more hands-on biology training of modern techniques into classrooms. And so that's kind of my general background so that you can have an idea of where I'm coming from for this deck. Tonight, what I'm going to be rolling you through is kind of where CAR T technology meets CRISPR, which is literally the last couple of years. So let me go ahead and share my slides. Share my screen, and then we'll get kind of rolling here. Shut all these other windows down really quick. Boom, boom. All right. So chimeric antigen receptor T-cells, CAR Ts, therapies, and their intersection with CRISPR. So with tonight's presentation, what we're going to look at is a bit of the history and things like that. But before we do that, let me just tell you who else is involved in kind of doing the role I have. So Lee Brown is also here. Lee is also a curriculum and training specialist for Bio-Rad in the central region of the United States. She's based out of Texas. Tamika Stubbs holds down the East Coast. Aaron holds down all the states in between. Everybody here has been a former classroom teacher. A lot of us have also had research experience. And so we've got kind of knowledge from both those areas, and that's what makes us a really dynamic team to kind of help you bring things into your classroom to kind of basically level up what you're doing with students. So this evening, what we're going to cover is more or less this outline. I'm going to take you through kind of chimeric antigen receptor therapy history, and then we'll kind of look at CAR T as CRISPR kind of intersects with it. And we'll specifically kind of dig into one paper that just came out within the last 6 months, which is basically just T-cell therapy completely CRISPR engineered, where previously a lot of things have been done using lentiviruses, retroviruses, zinc finger nucleases, and talons, a bunch of these old school editing, like second generation genome editing tools. And then if we've got time, I'll give you a little bit of a preview on Bio-Rad's CRISPR kit, because this allows you to understand a context under which the students might be interested in it, the CAR T stuff, and then a way to kind of teach CRISPR in the classroom really easily. And then we'll open it up for Q&A at the end. So CAR T, the elevator pitch I would give most people is it's a way to reprogram the immune system to recognize specific targets, usually cancer, and then take them out. So for as long as we've known about the immune system, we've been thinking, man, it'd be great if we could just get it to focus and really take out targets that it's having a hard time with. And CAR T is basically one of the best technologies we have for really doing that, really focusing a lot of the armaments of the immune system at things like cancer. The general workflow for most CAR T therapies looks like this. You're going to harvest T cells from the patient. You'll take them out, and in the laboratory, you're going to modify those T cells. And this comes through various different technologies. For the most part, recently it had been kind of lentiviruses, retrovirus work to put basically what we call a chimeric antigen receptor into these cells. More or less a way to get these cells to recognize specific antigens that we would want them to go after. These antigens tend to be antigens that are being presented on, say, a tumor cell, things like that. Grow up these T cells and then basically regraft them into the patient with the hope that these regrafted T cells get in there, find the tumor, and then tell the rest of the immune system, come here, let's beat this up, let's get rid of this thing. That's the general kind of schematic that most of these therapies fall into. So just know that that's in the background of all of this. I'm not going to dig super deep into every single step of the manufacturing or even the patient experience here, because those are all huge stories within themselves. So I'm just going to kind of give you the high points of those. For those of you that haven't thought about the immune system in a while, myself included, being a genomicist, let's just do a quick drill down of stuff that you need to think about in order to understand where CAR T therapies are going to fit in. So if we think about T cells in the immune system, they can recognize antigens that are presented to them by, say, antigen-presenting cells like dendritic cells. So a lot of us got an unfortunate crash course in this in the last 3 years due to a lot of our vaccines for SARS-CoV-2 being based upon this. Those T cells then, once they recognize an antigen, can then more or less initiate a downstream influence of immunoactivities, i.e. Triggering effector cells to go through interleukin secretions to get things like non-specific defense started. That would be your macrophages, your neutrophils, to get cellular immunity going, like killer T cells, and humoral immunity, like B cells that are going to produce IgG molecules that can then refocus more of the immune system on the target, and then also be able to produce on the side clonal selection to get memory T cells out of it. But the big picture here is trigger that T cell to then more or less kickstart all that stuff that goes on in the immune system to get it focused in on a target to remove it. For these T cells, what people have been hoping to do for years is basically reprogram them to be able to recognize specific targets on cells that either have a virus, that have a tumor, or are part of a tumor, et cetera. So when we talk about CAR T cells in a general format, this is what we're looking at. We're looking at taking portions of different known proteins involved in recognition and downstream signaling, and basically making a chimera of them that will allow a T cell to recognize an antigen and then start a specific cascade within the immune system. So there's usually an extracellular component, and this tends to be the variable regions, more or less, the heavy and light chain from an IgG molecule that are kind of stitched together so that they can act as a receptor on the outside of a T cell. Then you've got a transmembrane domain that's going to basically help stabilize it, hold it in place, and then a number of kind of signaling domains that then can be triggered when that extracellular region comes in contact with its antigen. And most of the different generations of CAR T are basically adding new signaling molecules into that intracellular region of the chimeric antigen receptor systems. So if we think about T cell receptors as kind of generations, and depending on which paper you read, there's everything from like 1 to 5, 1 to 6. And for the most part, when you read them, it's like everybody agrees, I think, on the first 3. And then every paper, 4 is more or less the same. 5, then there's a bunch of asterisks, and some of the papers are kind of calling different ones. So I'm just going to give you guys the first 3 because everyone seems to agree on those. And I think they're kind of instructive as far as what's going on in the general growth in this industry. So if we think about a normal kind of receptor on our T cell, that's what we have there at the left, this multi-component domain system that can recognize antigens and then cause signaling pathways. So these CARs, the chimeric antigen receptors that have been added, are definitely pared down versions of that. And more or less what we see throughout the generations is basically adding on of additional intracellular signaling mechanisms that then do things like increase the longevity of your T cell, increase the influence of that T cell on other parts of the immune system. So for the most part, first generation, you've got your variable region in order to be able to recognize an antigen, and you've got your CD3 zeta, which is a domain that you see associated with a normal T cell receptor. And that basically kind of triggers your normal intracellular T cell reactions to, hey, I saw an antigen, now I'm going to kind of trigger my normal T cell behavior. Second generation adds on CD28. Third generation adds on 4-1BB, and we'll kind of dig into those in a second. Our first generation really gets kickstarted in the 80s. So a lot of people, CAR T, they may have only heard about it in the last couple of years, but CAR T has been a field for a relatively long period of time. So the big kick, I would say, is with Dr. Kuwana's paper in 1987 out of Japan. And what he does is he takes the variable region from an IgG molecule that's an anti-phosphochlorine, and he basically makes this so that he can have it as a receptor on a T cell, and he adds this to an EL4 T lymphocyte. And basically he's able to show that, hey, I can add this receptor system onto T cells and I can get them to recognize the antigen in solution. And this more or less kind of starts opening the door for, hey, we could maybe be programming T cells to recognize very, very specific things. Dr. Arthur Weiss over at UCSF kind of takes that one step further. So he takes that ability now to build these receptors and adds that CD3 zeta system onto it, which basically gives you that normal intracellular signaling through the protein kinase cascade that leads to normal T cell responses to antigens that we saw within that first kind of immuno slide. CD just stands for cluster differentiation. So at this point, we've got a relatively simple system. We've got our ability to recognize an antigen. We've got something to hold us into the membrane, and then one intracellular domain to help us trigger kind of the rest of the cellular behaviors. These immediately kind of kickstart the first attempts to make a usable drug from these systems. So remember, that's 1991 that UCSF is working on this. And by the mid-90s, you have the first companies trying to go to clinical trials with it. Not too surprisingly, these companies are right across the bay from San Francisco. These are in Hayward, California, where Cell Genesis is based. And they basically had two that they went after in the mid-90s. The first one was actually to go after HIV. And it was both of these, I think, if I remember right, are phase 1 and phase 2 combined clinical trials. And the reason you see phase 1, phase 2 combined here is because the number of people you're going to enroll is relatively low because the cost is high and the process is big. And so that's why you see these kind of combined in here. And so they're going to use a retrovirus in order to basically set up your chimeric antigen receptor in these T cells and then program it to recognize, I think it was GP120 in the HIV one, and then try to harness the immune system to go in and clear cells that have this. And they end up trying to do a similar thing with colorectal cancer, but using an antigen that you see in these colorectal tumors. The bummer is none of these really get out of phase 1, phase 2. And it's not because of safety. So phase 1, remember, is your first checking to see, is it safe? Phase 2 in clinical trials is usually to show, is it effective? Both of these, they show to be safe. So there's no adverse effects that the patients go through, or no serious adverse effects the patients go through. But in both of these, you fail to get any better outcomes for the patient. So with HIV one, the grafts didn't take really well, and so those T cells just didn't hang around for very long. In the colorectal cancer one, you actually got a better grafting, but you didn't see better clinical outcomes. And not too surprising because we know kind of solid tumors are really hard to work with a whole bunch of different things, chemotherapies, et cetera. So what we get though out of this first generation is that we're at least on the right track to something. We've got something that's safe, but it's not quite yet effective yet. And the other side of this too is we've also demonstrated that you can do robust manufacturing around generating these. And that's kind of also a big deal because setting up a really big new process like this is not a minor thing. And so be able to demonstrate that you can get this major manufacturing system set up to do this and make a safe drug out of it is big in itself, even if none of these get to be a drug or a therapy that ends up in the market. So rolling out of the 90s, we kind of get our next big target that people are thinking about with CAR T, and this is mainly lymphoma. So the drugs that are, I would say, going to make the big splash all go after lymphoma, right? And if you think about lymphoma versus solid tumors, they're a lot easier to kind of get at. And we see this was the same for kind of chemotherapies, right? Chemotherapies did a lot better originally going after kind of bloodborne cancers. And you're going to see the same thing for these immunotherapies. So why go after lymphoma? Well, in the United States, you have 80,000 people that are diagnosed every year with non-Hodgkin's lymphoma. So there's a huge need. And in this same time era, a lot of primary research had been done to understand lymphomas in much, much greater detail, which really opens up the door for making really good CAR T therapies. Part of this drills down to kind of two major things. The identification of CD19 as a really fantastic target. And so it's a transmembrane glycoprotein that's found on normal and cancerous B cells, but it's not found on other tissues. And really importantly, it's not found on the stem cells that are leading to your immune system cells. And so you don't have to worry about those getting banged up if you're going after this as a target. And then also following up on this, basically an antibody that was really good at recognizing CD19, which was known as FMC63. And so this FMC63 IgG basically gets rearranged so they can become the chimeric antigen receptor that's in the intracellular domain for these therapies. And so these second generation ones, we're using that FMC63 so that we can recognize CD19 on these lymphoma cells. Then they've got their transmembrane segment, and then they've got new kind of switches or intracellular signaling in order to hopefully get different types of stimulation of the immune system and longevity of the T cells. So Cell Genesis is, of course, working on this. Man, how did those guys get so much funding? They were obviously just working on a bazillion things at once. Cell Tech was working on this, and they really kind of pioneered pushing CD28, which is an interleukin-2 kind of production trigger. It also helps with cytoskeleton reorganization of the T cells and kind of does a T cell anti-apoptosis pathway. So it should do a further triggering of the immune system, make your T cells hang around for longer. And so this one was a real good flagship, basically, domain to add on to your CAR system. And then in 2004, Dr. Mia at St. Jude's in Nashville really kind of brought to the forefront the use of 4-1BB, which basically greatly increases your survival rate of your T cells. So that way, if you do a graft, your T cells are going to hang around a lot longer, which hopefully is able to keep the immune system focused and targeted on your cancer for a longer period of time. These two different intracellular domains you see end up getting enrolled into two different drugs. And so we're going to cover those, because these are the ones that I think make the real big splash. So the first one is Chimera. And so a lot of people have probably at least have heard of this. And this is a drug that was developed by Novartis. And in 2017, it becomes the first FDA-approved CAR T therapy and really kind of the first gene therapy approved by the FDA. And it's approved as a third-line therapy for relapse refractory acute lymphoblastic leukemia. And so this means, as a patient, you have to have failed out of 2 other therapies before this is going to be available to you. But it was gangbuster, i.e., you had a really good clearance rate, high survival rate. So your survival rate on this ends up being about 60%. And this has been out there long enough now that there's really good studies on this. There's some really great papers that really dig into, how long do T cells persist in these patients and things like that. The only tricky point here is the cost of this, because these are autologous treatments, i.e., they are customized for every single individual, are really expensive. And so you're looking at $373,000 up to $475,000 for a treatment. It was such a blockbuster kind of therapy that they were able to go after, or apply for the FDA to go after, other diseases that this could be used for. So in 2018, they get approval as a third-line defense for diffuse large B-cell lymphoma. And in 2022, Chimera gets approved for follicular lymphoma. This drug is now, its drug/therapy, right, is available all over the place. And so within the United States, there's 163 centers now that one could go and get treatment using Chimera. I couldn't actually, I had a really hard time finding all the centers around the world, because Novartis does a really great job of looking at your IP and keeping you for only seeing this stuff in your country. But they've got manufacturing in 5 countries, which means they've got centers all over the place that you can go and get this treatment. Japan just came online. And so right now you basically have North America being covered, Europe being covered by 3 manufacturing locations, and now Asia being covered by the new manufacturing plant in Japan that just came on this past year. Manufacturing uses lentivirus, and in this case, in order to basically deliver your your CAR package to the T cells for the manufacturing. Right behind Chimera, and when I say right behind, we're talking a couple months after Chimera gets approved, another CAR T technology gets approved, and this is Yescarta by CART Pharmaceuticals, or CART Pharma, which is bought really quickly by Gilead, because Gilead doesn't want to fall behind on this. So they get approved in October, and if I remember right, Chimera came out in July. They get approved as a third line, once again, for large B-cell lymphoma. Also, really, really good stats on patient performance from the drug, really high survival rate, greater than 50%, much lower T-cell persistence, though. So this is one of the things that, if I had to choose between them and I had an option, I'd probably be going for Chimera, because I think that T-cell persistence really allows for better long-term protection. And then we have a similar kind of cost. But compared to some of the other things that are out there, because remember, all of these people that are getting access to these, right, are people that have not been able to respond to 2 other treatment types. So you've got a really resistant cancer already. So the fact that these are able to get you these really high survival rates, above 60 in the Chimera, above 50 here, means that these are really powerful tools at fighting these cancers. And if they were cheaper, we'd probably be not using them just as third line, but as second line or maybe even first line. 2022 is really interesting because FDA approves Yescarta for second line for diffuse large B-cell lymphoma. And I'm not quite sure all of the reasons behind that. I'm not sure if it's because of difficulties with the other technologies for treating that, or if it's because of costs being pushed down. But this makes me optimistic that these costs might go down in the future, which would expand the number of patients that have access to them. And maybe these get scheduled to be more second line and maybe down the road, first line technologies. Yescarta is using the CD28 instead of the 41BB that Chimera is using. And so I think that's what a little bit is leading to this T-cell persistence being a little bit lower. This one, just like Chimera, is accessible all across the United States. There's over 110 treatment centers now that do this. And the reason I'm calling out those treatment centers, because if you remember the beginning there, I showed you kind of the general workflow. This isn't like a simple, like, go in and get an injection type of thing. Like, this is go in, get apheresis to get your T cells out. Those T cells have to go out someplace to then be manufactured, come back, and then get grafted onto you. And so that's why the treatment centers thing is a really big deal, because these are big protocols that you have to be able to execute at a center to know that this therapy is going to work. Right now, Yescarta only has kind of 2 or 3 manufacturing locations, 2 in the United States, 1 in Oceanside, California, where Kite was originally first doing this work, and then 1 in Baltimore. And then they've got 1 in Europe now for the Netherlands. Their modification system is a MSGV retrovirus system. And then if you're looking at those and you're like, well, why wouldn't you just use both those intracellular domains to make a super drug? Well, it's because it just took time to kind of get that built and then into the FDA pipeline. And so we do see that, and that's what people would call kind of your generation 3 CAR T. And so this is, probably going to butcher how you say this, Breyanzi, which is from Juno Therapeutics. In 2021 is when this gets FDA approval as a third line treatment, once again, for large B-cell lymphoma. Kind of what you would expect based upon the stats from the past 2. You've got a really high survival rate, greater than 50%. T-cell persistence, we still don't have a great look on that yet, just because this hasn't been on the market as long as those other 2. But cost is in that same ballpark, and that's probably what's going to keep this in the third line group for a while, is when you're saying it's going to cost $500,000 for a treatment, a lot of people are kind of going to delay that and keep that where it's a little bit harder to access. And also, it's not just the price. I mean, the effort that goes into making these is not inconsequential, because you've got basically a 30-day work cycle in order to make these systems work. You've got to be able to harvest T cells from the patient, do this really complex manufacturing on the backside, and then regraft it into the patient. It's really, really complicated, especially compared to some of the kind of frontline things like chemotherapy, right? I've got a drug that's on the shelf, and we're just going to infuse you with it. Or some of the second lines, which is, we've got a drug that's now been coupled to an antibody that can recognize antigens that we usually see on these type of tumors. And so the difference between allogenic, basically versions of drugs, things that can be off the shelf because they can go to anybody, versus autologous, is what kind of keeps these prices high and probably keeps them in the third line for a while. Breyanzi or Breyanzi can be had now in 84 different centers across the United States. Most of these are, of course, the same centers that are offering those other three, because they've got these skilled workers now there to be able to do that. And they only have 2 manufacturing locations right now, one in Bethel, Washington, and one in Devens, Massachusetts. But from reading kind of some of the business literature, they're trying to get one up and going in the European Union at this point. They're also using lentivirus in order to do their manufacturing. Let me give you a zoom out of what the kind of process looks like for building the therapy, and a little bit of like what it looks like for getting into the patient at these centers. This easily could be a talk within itself, because the manufacturing of the viral vector systems is relatively complicated. I actually kind of deleted out 3 other slides. I was like, we're just getting way too into the weeds, especially because I think some of this is going to go away in the short term as CRISPR takes over the work that was being done by these viral vector processes. But in general, let's just kind of cover it so you understand some of that complexity that's going on in the background. You're going to be growing up cells in order to basically transfect with your viral vector system. You'll then be harvesting viral vectors from there, capturing them, polishing them, and you then can end up with this kind of finished vector system that more or less has the chimeric antigen receptor that you're going to want to deliver to the patient's T cells. And so if you look through some of these businesses, it's kind of interesting because some of them actually don't do this part. They contract out with somebody else to do it. In fact, there's a huge business in Houston that does this for a handful of those other companies. So those other companies are basically taking this viral vector that's been kind of processed for them from this other company, and then they're doing this manufacturing, which basically starts after they get the apheresis material from the patient at the hospital. So the patient gets apheresis done, T cells basically get enriched from there, they get activated, and then transduction takes place at the manufacturing of these companies. That transduction is using that viral vector to then drive in the chimeric antigen receptor into those T cells. So now you've got these T cells that can recognize a very specific antigen for those patients in cells that are theirs. They expand those at their facilities, and the expansion has been a really interesting thing because a lot of the original clinical trials, as they're building up toward that, this was a big struggle point. So there was a lot of innovation and expansion of these that took place. Those, once they've been expanded enough to get to a high enough titer, they can be cryopreserved, sent back out to the treatment center where they are then grafted back into the patient through an IV. And so because it's a really complicated system, there's a lot of time, a lot of money that goes into it, and that's what keeps these prices kind of really high, and which makes these, you know, kind of like a big deal if somebody's getting this done. Like I said, these are month-long processes from when a patient is aware they want to get the treatment to when they are getting the treatment, and that's because they're getting apheretic material done from them, manufacturing's going on the backside once they understand what the patient's going to look like, things like that. So a lot happens for these, but I think we might not have to worry about this system as much, because as CRISPR moves into this market for modifying T cells, I think this is going to go away. So these technologies were groundbreaking, right? They are getting people treatment for diseases that were not able to be treated, right? And that's why they were third line. But there's still a lot of room for improvement on them, because a lot of these treatments also have some negative effects that are taking place during the treatment process. A lot of patients end up with cytokine release syndrome. So you've got a bunch of T cells you've given somebody, right? And you've given them a target. They all show up at once, and you have this giant explosion of cytokines that get produced, right? So the patient goes through this huge swarm of basically having your immune system get really, really amped up for a short period of time, which can have all kinds of negative effects. One of them can be neurotoxicity. Some of the treatments for the neurotoxicity that they've seen with these treatments has just been steroidal treatments for, about a handful of days to a week. And this seems to usually calm most of these down. Neither one of these are so bad that people are avoiding these technologies. Otherwise, the FDA, right, wouldn't have approved these. Also, the other big room for improvement here is you're talking 20 to 30 days from when the patient wants to do this to where they can actually get the treatment. This might be able to be sped up if this isn't as autologous work, individualized work, and we could get some allogenic products out there that are more or less off the shelf. One way to do this would be able to make allogenic T cells so that you could have a big bank of T cells that you could just quickly modify for the patient and be able to quickly graft them. And then also the other thing, and this is partly due to the autologous nature of this, is the cost in all of these is really high. And then there's one other thing that to some degree kind of trickles our way into what's going on with CRISPR moving into this environment. And this is that those T cells you've harvested are T cells that still have their endogenous T cell receptor on them, which means you've got a T cell that you've now focused a little bit, right? You've given it an antigen to say, hey, I want you to look at this, but it still has its old T cell receptor on there, which means it can be a little bit easily distracted. And so it'd be great if you could further focus this cell, right, by getting rid of that T cell receptor. And how could you do this? Well, you could go in, right, and potentially disrupt the genes involved in making the endogenous T cell receptor. And the technologies previously available for doing this, these genome editing technologies like zinc finger nucleases or talons, are not super easy to work with. And so they don't really fit into a pipeline really well where you could do this. However, within just the past decade, right, or even just like the past 5 years, CRISPR has just grown to be a powerhouse when it comes to a genome editing tool. And that's because it has really high precision, it's cheap and easy, really, really easy to use compared to zinc finger nucleases and talons. And so if you aren't familiar with CRISPR, I'm just going to give you a really high-level view of it, and then we'll kind of dive into how it's being used here with T-cell reprogramming. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and this harkens back to how we originally kind of found this system in bacteria, because we were seeing clustered regularly interspaced short palindromic repeats way back when. We weren't really sure what they were doing. And then within the past 15 years, people realized that this was an adaptive immune system. And people realized how to basically turn this adaptive immune system into a technology we could use to easily and quickly edit genomes. Associated with CRISPR, you'll often see the word Cas, and all a Cas is a CRISPR-associated protein. For the most part, these tend to be nucleases. In fact, most of the time when you hear CRISPR in the last couple of years, it's been CRISPR-Cas9. And the elevator pitch for this is it's a programmable restriction enzyme. And when I say restriction enzyme, I don't mean like EcoR1 with 6 nucleotides. If you're going to go try and cut in a human genome, you're going to cut 80,000 times. It's programmable with a 20 nucleotide search string. And that gives you high precision within something the size of a human genome. In fact, this is longer than a lot of PCR primers that we use to do work within humans. So the way the system generally works is this. You've got the nuclease, Cas9, and you can dock something known as a guide RNA into it. Basically, this is an RNA molecule that then has this 20 nucleotide sequence attached to it that you can program so that you can go anywhere in the genome and look for this 20 nucleotide sequence. If you can find that complementary sequence, what will happen is that Cas9 will hold down and it'll give you a double-strand break right there. If you can do that, you've basically got the capacity then to edit the genome, right? Because if you can do a double-stranded break, you could delete stuff, you could stick stuff in there using homology-directed repair. There's all kinds of things you could do. And so there's a lot more we could go into with CRISPR, but I'm not going to do that now. All you need to know is it is basically a programmable restriction enzyme that can allow you to go in and cheaply, easily edit genomes. Where this interfaces with CAR T technology comes through really strongly in this 2020 paper. So this group basically is running something similar to what those CAR T protocols we looked like before in those other drugs, except they're going, hey, you know what? We want to focus that T cell even more. So let's get rid of that endogenous TCR, or the endogenous T cell receptor. And so what they do is they harvest the T cells, they take the CRISPR-Cas9 system, and they basically give it a program, 20 nucleotides, that says, hey, here's some genes in the TCR. Why don't you go in there and cut it up for me? So CRISPR-Cas9 goes in there, and it basically gives you an interruption so that you can no longer make that T cell, that endogenous T cell receptor. And they're like, you know what? We're already in here. We want to modify one other thing. So PD-1 is a receptor that's involved in basically programmed cell death. And so if you can kind of get rid of PD-1, you have probably a T cell that's going to be able to hang out for a lot longer, because it will never basically be able to get a signal that says, hey, you know what, I need you to die now. And so they modify these T cells using CRISPR-Cas9 to knock out basically those two systems. And then they do their typical, not typical, but, you know, what we've become typical CAR T technology, which is you take a lentivirus and use that to drive in a cassette that contains a receptor system that you want to use. And so they deliver this new receptor system. And now you've got these T cells that don't have the endogenous TCR, they don't have that PD-1, and they've got a receptor system you've developed that looks for a specific antigen. And they basically regraft this into the patient. And this is great because now we've got a more focused system with longer lifespan T cells. The only bummer is they did just add a ton more complexity to this, right? They just added a whole nother manufacturing step in here. And you've got your old CAR T manufacturing, which was like a beast to begin with. And so this paper kind of is, I would say, the in-between points between classic CAR T therapies and what's going to be the CRISPR generation of T-cell modified systems with CRISPR. And that full kind of like, let's move all to CRISPR happens within the past year. So this paper that just came out, this non-viral precision T-cell receptor replacement for personalized cell therapy, is so dense, I had to go through it 8 times. And so I've just tried to distill it down to the kind of points that make it very obvious on how CRISPR is going to be the thing that we go forward with, and we will probably leave a lot of these lentiviruses and retroviruses in the past in order to make T cells that can focus the immune system on specific targets like cancers. So the way that their system works at a really, really high level is they're going to take T cells from the patient, and they're going to they've developed a CRISPR-Cas9 system that will go in, and it's going to cut the genome right in the middle of a bunch of the genes that are involved in making the endogenous T cell receptor. So they're going to basically knock up what ends up making the alpha and beta units of the T cell receptor. So they cut the genome right there. And when they've cut it, they now have the ability now to modify the genome by putting something in there. And so they'll do homology-directed repair. And in that cut site, they're going to put in new alpha and beta units that are for antigens that they've selected for. And their antigen selection I'll get to in a bit, because that's a whole nother ballgame in and of itself that's very impressive. But basically at the end of this, what you have now is you've got a T cell that doesn't have the endogenous T cell receptor. It has one you've completely engineered with very, very well chosen receptors. The beauty about having it not be that old school chimeric antigen receptor where you're kind of slowly tricking out intracellular systems is you've now got all of the hardware that evolution built working for you, because all you're doing is switching out those 2 domains and letting the rest of the system work as it normally does. So you've got this hyper-focused, fully outfitted T cell now. The way they get to this is really impressive. So what they'll do is from the patient, they do a biopsy of the cancer, and the incredible thing here too is they're going after solid tumors. They're not going after blood cancers. So they're doing a biopsy from the solid, they're going after a blood draw, They're using sequencing to then predict and prioritize epitopes, basically the little piece, right, of an antigen where the antibody really recognizes it. And they're generating these neoepitope HLA libraries from that. They will then take T cells they harvest from the patient, and they will use that library now to capture T cells that can recognize that epitope that they think is going to cause a really strong stimulation of the immune system. Once they've captured those, they can then pull out the sequence from those cells to realize that is how that cell is making the receptors. And then that sequence is going to be what's used for their CRISPR system to build those new T cells. And so what you're basically doing is sorting through the potential receptors the patient could be making. They would be hyper-focused to find that tumor antigen, and then just concentrating those into T cells. Once they've done a selection there, and they're basically, for this right now, they were just selecting 3 of the best kind of epitopes for this. They then go through and they can do manufacturing for the patient at a larger scale. So they'll do apheresis of the patient. You'll collect a bunch of T cells, use your knowledge of your screening from your last piece to then take those T cells, modify them to give them just the receptors you want, get rid of the endogenous T cell receptor, expand the cells, and then regraft them into the patient. And you get this potential now for this super targeted, super focused T cell that could potentially make a big difference on tumors. So the outcome of this paper, we've only got 16 folks that make it through this phase 1, phase 2 clinical trial on solid tumors. And remember, these are people that failed 2 previous types of therapies. So this is a third line one once again. What we can say is that for sure, this is a safe technology. We have 1 cytokine release syndrome within this 16 group, and 1 person that had a meningitis-like effect from it. But this person had a history of kind of neurological issues with meningitis. Both these were treatable with known technologies. Is it effective? Well, in some of these, they did see tumors decrease in size. Some, they didn't see much change. But what they can definitely say is that in all of these tumor tissues, they were able to find these T cells that were modified in there, stimulating the immune system. And this is within the first 30 days from the graft. And so what we think is that there is a really big potential here, just like how kind of that first generation CAR T, right? It was safe, but it didn't necessarily knock the cancers out. I think we're at the same doorstep here, where now we've got this system that we know is safe, and being able to kind of expand upon it is going to probably get us to generation 2s of these that will potentially get rid of, say, something like a solid state tumor, which would be an amazing, amazing breakthrough. So the big upsides on this is there's no virus manufacturing pipeline. So there's some savings on the time and money. We've got a big speed here because we're only having to go through and grow these modified T cells and do the Cas9 production in the background, which is a lot faster than that whole virus production manufacturing pipeline. The ease, so it's really easy to basically add more of these neo-TCRs, more of these potential targets because of the way CRISPR systems can be built. So right now they're just using 3, but maybe you decide to add more onto that, which gives you a better potential for harnessing more of the immune system come in and see it. And it's got a really good focus, right? You don't have that endogenous TCR there distracting your T cell. So there's some really, really big upsides that I think are going to open the door for this kind of second generation CRISPR modified T cell therapies. So I think in the future, what we're going to see is an increase in the speed and a decrease in the cost as CRISPR becomes the big player in T cell modification. We might see allogenic off-the-shelf ones be produced, I would say, in the next 5 to 10 years. I think there are some of those when I dig into some of the clinical trials. And another big thing is CRISPR production is a lot easier to automate than some of the lentivirus and retrovirus pipelines. And so I think there's also potential of just speed and decrease in cost, even for autologous solutions through automation of manufacturing. And being somebody that worked in genomics, I can tell you, automation makes a big difference. When we work on the Human Genome Project, we're using the same sequencing technologies, the ABI 3730, but it was our automation that really started crashing the cost of base pair sequencing. And I think we could see the same thing when it comes to automation of CRISPR-related manufacturing. So, and I think the underlining one there in all of this is that CRISPR is going to become the dominant tool in T-cell therapies. And in fact, the easiest way to see that is go over to clinicaltrials.gov right now, type in CAR T. You can see all the ones that have been in the pipeline and the ones that are there. And there's quite a few of them. There's 1,200 of these in clinicaltrials.gov, which is a pretty sizable number for a lot of other things you might look up over there. And if you type in CAR T CRISPR, you can see there's about 20 in there right now. But I think this will explode, especially after that paper just came out, right, within the last 6 months. Because a lot of people look at that and say, hey, the door's now open. Let's get rid of our lentivirus or retrovirus systems. Go for CRISPR as the way to modify these T cells. And so I think the future is really bright in that regards. So that's more or less kind of what's going on in the CAR T meets CRISPR world. If you're trying to figure out a way to bring in a hands-on way to kind of at least allow your students to understand CRISPR, Bio-Rad does have a kit that allows you to do that. And compared to that paper, a cool thing here is your students are going to do a relatively similar thing to what they're doing there. You're not going to be working with T cells in your classroom unless you already are. If you are, let me know. That would be really cool to see. But what you will be doing is you'll be using CRISPR-Cas9 to do a directed cut and then use homology-directed repair to then stitch something into the genome. Instead of stitching in, a new T-cell receptor, all we're going to do in this case is we're going to stitch in a stop codon so that we can basically stop the production of a gene that we're using in order to get a pigment outcome that allows us to tell if we have a functional LAC-Z gene or not. So in general, this is what the process looks like to the students. They start off with cells with a functional LAC-Z gene, and if you're using XGAL in the auger, you have a blue pigment due to that. After doing CRISPR to make a cut and do HDR, you've dropped a stop codon into LAC-Z, which stops it from functioning. You can no longer metabolize XGAL, and you'll see white cells that allow you to say, yep, I have gone through and I've definitely disrupted that gene. And so it allows you to really, I think, dig into the big portion of CRISPR, which is the ability to kind of do more of the synthetic biology stuff, not just go through and disrupt stuff, but be able to go through and add stuff using HDR, which is going to be where CRISPR, I think, really excels over some of the other mutagenesis and things like that we've had out there for a while. Let me see how we're on time. We're about 10 minutes in. What I might do is this. I might just give you a quick overview of this, and then we'll jump into questions. So that way, if you guys have questions about what was going on in CAR T, we can get to them. So in general, like I said, the Bio-Rad kit, what you're going to do is you're going to use HDR, CRISPR-Cas9 to go do a directed cut, drop in a stop codon into the middle of that. It is a really well-built kit in that it has a number of controls that allow students to interpret that what's going on is actually CRISPR-Cas9 and not just some hocus pocus where you've given them a different vector that is producing a different pigment. And so this relies upon kind of a handful of conditions with the system. The bacteria we're using, E. coli, if they get a double-stranded break, they are basically knocked out. They have a lethality due to that because bacteria usually don't have the ability to repair a double-stranded break, or at least E. coli don't. We also have Cas9 that is being continually expressed in these E. coli cells. And we've got a repair machinery for those bacteria we can turn on and off. And so we can actually make bacteria that can repair themselves when the native ones can't. And so what will happen is students will take bacteria that have the repair mechanism turned on, bacteria that don't have the repair mechanism turned on, and they will use a bacterial transformation to drive plasmids into them that contain different components of the CRISPR system. So on our first two plates on the left here, these are coming out of starter colonies where the E. coli do not have the repair machinery turned on. And in the first vial there, or the first kind of A sample, the students are going to deliver a plasmid that just has donor DNA. So this is donor DNA for doing HDR, homology direct repair. But in this case, you don't have a guide RNA for the Cas9 to use to cut. And so what happens is, even though you have donor DNA there, you can't deliver a cut into the LAC-Z gene. And so these bacteria end up not being modified at all. They'll look just like how the student started. They'll end up with blue colonies. If we move over to our second tube here, our B, we now have the students moving in a plasmid that has guide RNA that can target LAC-Z, and it has the donor DNA for the stop codon. In this case, though, they're starting with bacteria that don't have the ability to repair. And so when Cas9 grabs the guide RNA, it'll do a double-stranded cut, and the bacteria can't recover from that. And so what the students will see is they'll end up with no growth. And so this tells them, hey, I know that I had a cut that took place there because my bacteria didn't recover. If we move to our second kind of starter plate, this now is a lineage of bacteria where we've turned on a repair mechanism system, and we're going to transform them with the same two plasmids from the first set. So in C there, we're going to move in the plasmid that has just the donor DNA. What happens is there's no guide RNA, so you can't get a cut. And so just like plate A, the students are going to see the bacteria basically have not been modified. They're just blue. When we go to kind of vial D there, you've now have the plasmid. It's got the guide RNA and the donor DNA. So the guide RNA will be picked up by Cas9. You'll get a cut in LAC-Z. And since that donor DNA is there and the repair machinery is turned on, that repair machinery will grab that donor DNA, use it for homology-directed repair, and then stitch in a stop codon. And what they end up with is white colonies, because with LAC-Z not working, you can't get xgal to be broken down, and so you just have normal-looking white E. coli colonies. Those controls are really important because that's what allows them to interpret that you haven't just given them some fancy plasmid that just is now producing so much white pigment that they can't see blue pigment. And so you can use these controls to basically show them, yes, what you've really done here is CRISPR and not just some hocus pocus. And ultimately, this is what their results look like from that series. And then if you want to, you can take this even further and you can do a genotyping extension with them where they can take those colonies, use multiplex PCR to diagnose that a stop codon has been dropped in there. I won't dig too far into that. Just know that it's a multiplex PCR. They'll be able to interpret gel results and say, yep, what I've done is I've placed a stop codon, or I've at least placed DNA in the middle of my LAC-Z gene, and that's what's causing the interruption. So I'll go ahead and stop there. So that way, if there are questions, and then I think, Lee, you've got a slide you want to throw up as well for a questionnaire. And I will open up the floor to any questions that people have. And so if you've got a question, you can go off of mute and ask it, or you can put it in the chat and I can try and dig it out from there.
Hello, yeah, I got a question from
Yes.
Costa Rica. My question is, what happens with the off-target effects of CRISPR and this technology and CAR-T?
Yeah, so I mean, that's always been a consideration for all of CRISPR, right? And so with most of the CRISPR systems that are being used for the CAR-T, it's already going through a lot of the bioinformatic checks to be able to make sure that you're most likely not going to have a system that's going to deliver off-target cutting. And then in most of these systems, they're actually doing sequencing of those individual cells to make sure there isn't any off-target cuttings going on. So that last paper I showed you guys, the one that just came out 6 months ago, they go through and they do full genome sequencing of those T cells to make sure you haven't had any other modifications take place. So that also is an interesting thing, because that 2022 paper, it also really harkens to the fact that sequencing now has become so cheap that it just becomes another tool that you have to make sure the entire system that you've made looks like what you want.
Okay, perfect. Thank you so much.
Yep. Other questions? We do have a webinar survey that you can QR code from your screen right now that can help us understand what things may be helpful, what else we could be doing to kind of support you. And if webinars like this are helpful for you. And if you do have questions that come up later, you can always email us at explore@biorad.com, and those will come to me and I will do my best to answer those for you. I mean, I think one of the interesting things right now is that, I mean, most of us, unfortunately, probably have had somebody that we know close that's gone through cancer treatment, is that compared to chemo, I think that these technologies, when they become cheaper and more accessible, is also a little bit easier on the patient, on top of getting better clearance. Remember, chemo has been around for a really, really long time. And for the most part, it's a pretty crude tool. The tool's gotten a little bit better in the last couple of decades as we've more or less paired immunotherapies with chemo, i.e. using antibodies and things like that to direct chemo agents to your targets. But it's still a pretty broad, tool where these T-cell mediated therapies like this are a lot more precise. And I think we're really just at the starting place for these. So it's going to be a really exciting next couple decades when it comes to our potential to treat cancers. And especially being somebody that just had a family member, my father just went through treatment for lung cancer recently. I can tell you most of the tools that were used were pretty crude, pretty broad, pretty hard on the patient. And so having this as a perspective in the future is really, really welcome.